Hello everybody, this is Dr. Nadeem and we are with Neelam Path Lectures, the Pursue series. And as you must be aware that all our lectures are available on YouTube. We have a Telegram group which you can join to get all the information regarding our activities. We have a Google Drive where all the PDF of all the lectures are available. These are the disclaimers, and we are in phase 3, which is a series of recorded pathology lectures. And today we are with Pursue 23F, which is genetic disorder, and we are streaming from AIIMS Bhubaneswar, India. And to talk on genetic disorders and to take a series of lectures on genetic disorders, we have Dr. Amit Kumar Adhya, who is an MD from PGIMER-Chandigarh. Presently, he is a professor in the Department of Pathology and Lab Medicine at the AIIMS Bhubaneswar. He has also been the ex-professor of Kalinga Institute of Medical Sciences, Bhubaneswar. He's got 118 publications in international and national journals, 1 book chapter, 6 funded projects. With 21 years of teaching experience in students as well as research experience, he has completed 5 collaborative research projects funded by DBT and BRNS. He was also awarded the Best Paper Award in the IAPM 2005. Dr. Adhya will be taking a series of lectures on the basic things on genetic disorders, and the first session is on fundamentals of genetic disorder. So I would request Dr. Amit Adhya to start his lecture. Thank you so much.
Hello students, in this chapter I shall talk about the fundamentals of genetic disorders. We all know that the human cell contains 23 pairs of chromosomes, 22 pairs of autosomes and 1 pair of sex chromosome. The sex chromosomes are XX in the female and XY in the male. If you look at the cell, you will find that within the nucleus there are some dark areas and there are some light areas. The dark areas, as shown in this picture, are known as heterochromatin and the light areas are known as euchromatin. At the molecular level, the heterochromatin is basically the DNA which is tightly packed. The DNA is normally wound over a protein known as the histone protein. These proteins and DNA forms a complex known as the nucleosome. When the nucleosomes are tightly packed together, it is called as heterochromatin and it appears dark. When it is lightly packed or it is not coiled over each other, it forms a light area which is known as the euchromatin. The important thing to know about this euchromatin and heterochromatin is that the heterochromatin is inactive. It does not transcribe into mRNAs, whereas the euchromatin is the area from which mRNAs are produced. The mRNA is usually produced in the form of a precursor mRNA, which contains the exons, introns, and the promoter regions. After splicing, that is removal of the introns, the final mRNA is produced. The final mRNA contains an open reading frame and it is flanked on both sides by untranslated regions, UTRs. The final translation occurs only from the open reading frame and the UTR is not translated into a protein. The nucleosome is basically a complex of the histone protein at the center which forms the core and the DNA which is wound over the histone protein. The histone protein is a octamer and it contains various parts. A DNA segment of about 150 base pairs is usually wound over the histone protein and it contains 1.8 turns of the DNA material. In between two histone or bound DNA, the intervening areas are loose and they contains another type of histone protein known as the linker histone proteins. The genome is composed of 3.2 billion base pairs, out of which there are 20,000 protein encoding genes. This comprises only 1.5% of the genome. Rest of the genome, that is 98.5% of the genome, does not encode for protein, and hence until now it was known as the dark matter of the genome or the non-coding areas. However, the importance of these non-coding areas are being gradually unveiled, and we know that they are very important in regulation of the gene function, although they themselves do not produce any protein. The non-coding protein sequences are present within the genome, which account for 98.5%, as I have already mentioned. This usually consists of various areas in the DNA sequence known as the promoters and enhancers. The promoters are usually sites where the transcription factor bind, and enhancers are sequences which help in binding of the transcription factor to the promoters. Then there are these non-coding regulatory RNAs, which are of two types, the microRNAs and the long non-coding RNAs. We'll talk about them later on. There are certain DNA sequences within the DNA which do not have a fixed position. They are known as the jumping genes or the transposons. The function of these jumping genes is not known. The centromeres are the central areas of the chromosomes which act as tethers, and during mitosis they help in tethering of the two chromatids. The telomeres occur at the ends of the chromosome and they act as cellular clock. You might have already learnt about telomeres and the cellular aging in the chapter of inflammation and cell death. As I already mentioned, the important functions of these non-coding regions are regulation of gene expression. They act as the blueprint of the genome in the sense that they provide the architectural planning for the cells. Polymorphism refers to the difference in the DNA sequence in different individuals. You will be surprised to know that 99.5% of DNA of any two individuals is exactly same. It's only 0.5% of the genome which distinguishes from one individual from the other. But this 0.5% of the genome is equal to about 15 million base pairs. Another interesting fact is that we are 99% identical in terms of DNA of the chimpanzee. Now, what makes two individuals different from each other? The answer to this question lies in three things: the single nucleotide polymorphism, copy number variation, and the epigenetic changes. Single nucleotide polymorphism, as the name suggests, these are different nucleotides present at the same location in different individuals. As depicted in this picture, you can see there are three individuals, and at the same site the first person has adenosine, whereas the second person has guanine, and the third person has thymidine. So there is a difference in, say, single nucleotide at a given point. Such single nucleotide differences are many, about 1000 to 2000 SNPs are present in any two given individual. More importantly, these single nucleotide polymorphism or variations can occur in either the non-coding sequences or the coding sequences or in the intervening sequences. If they are present in the non-coding sequences, they do not produce much effect, but when they are present in the coding sequences, they can give rise to different types of proteins in different individuals. These proteins may have similar or dissimilar function, making the two individuals distinct. They can also be seen in the non-coding regions, such as the promoter regions. If these variations are present in the promoter region, then the gene expression in different individuals will vary, and it will make the two individuals different from each other. Now, importance of knowing SNPs is that it can explain or predict the difference in susceptibility of individuals for getting different diseases. For example, the same disease can affect 10 individuals, but all of them may not be susceptible to the disease. One person may be more susceptible than the other. Some person may express the disease in a different way from the rest of the persons. The explanation behind this could be due to the difference in the single nucleotide which is present in different locations in the genes. Another important thing that have come up recently is the copy number variation. As the picture shows, different persons may have different copies of the same gene. Increase in number of copies may lead to increase in activity of a particular protein, thereby altering the metabolism and catabolism in the individual. So the individuals, as shown in this picture, having less number of copies may be more obese. The individual having more number of copies of the same gene may be lean and thin. These CNVs have been found to be continuous stretches in the DNA material, and they can vary from 1000 base pairs to million base pairs. In any two given individual, at an average 5 to 24 million base pair difference in copy number variations can be seen. More importantly, the CNVs can occur within the coding regions, thereby altering the protein morphology and the protein functionality. Another important thing that has come up recently are the epigenetic changes. As the name suggests, epi means above. So literally it means changes which are above the genetic level. By definition, epigenetic changes can be termed as heritable chemical modifications of the DNA or chromatin that does not alter the DNA sequence itself. That means the DNA sequence, the adenosine, guanosine, thymidine, the DNA sequence is not changed. Rather, certain chemical additions and substitutions in the side chain of the DNA occur, and these changes lead to major phenotypic change in expression, and such changes are heritable also. Like genetic changes, they are heritable. Now, what is the significance? Significance is that epigenetic changes are found to have a lot of effect in regulation of tissue specific gene expression, X chromosome inactivation, imprinting, and the cellular mechanism of aging and cancer. Have you ever wondered why two cells have different functions in the body? For example, the beta cells of the pancreas produce insulin, whereas the skin cells cannot, although both the cells have the same sequence of DNA. The explanation behind this lies in the epigenetic changes. The epigenetic changes enable the beta cells of the pancreas to produce insulin, whereas the same genes are masked in the skin cells due to epigenetic or chemical modification, and hence insulin production is not feasible in the skin cells. That's why I have said the epigenetic changes are essential for regulation of tissue specific gene expression. X chromosome inactivation usually occurs during the fetal life. In the females, there are two X chromosomes, one of which is randomly inactivated and appears as a bar body. The X chromosome inactivation requires epigenetic changes. Imprinting. This is a phenomenon where one allele of a particular phenotype is inactivated. Normally there are two alleles for each phenotype, and both may be active or one of them may be active and the other inactive. The inactivity of this allele is basically attributed to this epigenetic changes. Other things in which epigenetic changes take a very important role are aging and cancer. In fact, the discovery that cancer is due to epigenetic changes have opened up many research areas because epigenetic changes can be targeted for therapy. Two things are there in the epigenetic changes: the chromatin remodeling complexes and the chromatin writer complexes. The chromatin remodeling, as the name suggests, leads to remodeling of the chromatin by shifting the position of the nucleosome. We have seen previously that the nucleosomes are structures where the histone protein has got a DNA wrapped around it, and the nucleosome is transcriptionally inactive. So if the histone protein shifts from one place to another, that will lead to inactivation of another set of genes and activation of some set of genes. This particular phenomenon is brought about by epigenetic changes and is known as chromatin remodeling. Chromatin writer complexes, these are enzymes which lead to chemical modification within the histone. Such chemical modifications are known as marks. The chemical modification may be in the form of either acetylation, methylation, or phosphorylation. Acetylation usually occurs in the lysine residues of the histone protein, and it is brought about by the enzyme histone acetylase. These are reversible processes because there is another enzyme known as histone deacetylase which can erase or remove this acetylation from the histone. Methylation of the histone usually occurs at the arginine or lysine, and the methylator enzymes are present. Phosphorylation usually occurs of the serine residue. Apart from the changes, chemical changes that I have discussed in the histone, chemical changes can also occur in the DNA itself, and that is known as DNA methylation. DNA methylation is very important because it leads to transcriptional silencing of the DNA. That is, the portion of the DNA which is methylated can no longer produce mRNAs. This picture tells you the same thing. The histone protein is at the center and the coil of DNA is all around it. Methylation or acetylation of the DNA or the histone leads to change in the shape of the nucleosomes, that is shifting of the nucleosomes from one place to another, thereby opening up some DNA portions. These open DNA portions are now able to transcribe. So epigenetic modification by changing the position of nucleosomes can lead to transcriptional activation or inactivation. Epigenetic changes have assumed a lot of importance nowadays, knowing the fact that many cancers have epigenetic changes. Epigenetic changes are reversible, and these epigenetic changes can be targeted. There are drugs available such as the histone deacetylase inhibitors which can reverse the process of acetylation and thereby treat cancer. Similarly, DNA methylator inhibitors are also available. You can search in the internet and Google about this kind of therapies. Another form of non-coding region are the non-coding RNAs. There are two types of non-coding RNAs. Non-coding means they do not transcribe for any proteins. They are produced from the DNA by the process of transcription, but these mRNAs, they do not translate into proteins. There are two types, the microRNAs and the long non-coding RNAs. The microRNAs on an average are about 22 base pair, hence their name is microRNA, whereas long non-coding RNAs usually are more than 200 base pair long. MicroRNAs are very important because they lead to post-transcriptional silencing of the gene expression by altering the messenger RNAs. In fact, this discovery was so important that in 2006 the Nobel Prize in Medicine and Physiology was awarded to the person who discovered microRNAs. Approximately 1000 different types of microRNAs have been encoded. In contrast, long non-coding RNAs are a set of RNAs which are usually more than 200 base pair long. About 10,000 to 20,000 LNC RNAs have been encoded. The primary function is gene repression, methylation or gene activation. They have assumed importance nowadays because it has been found that they have very important role to play in the pathogenesis of atherosclerosis and cancer. You will learn more about these things when we'll talk about cancer and atherosclerosis. This is a diagram to show you how the microRNAs are produced and how they function. The microRNA gene is present in the DNA and it is produced as a primary microRNA. The primary microRNA is transported into the cytoplasm where it forms the pre-microRNA. Here it engages with an enzyme known as the dicer enzyme. The dicer enzyme cuts the pre-microRNA into its component RNA, then it is called a microRNA. The microRNA breaks into its two strands. One strand having a nick at the center forms an imperfect match with the DNA. This imperfect match leads to suppression or repression of translation and hence there is no protein production of the mRNA, whereas the other end, which forms a perfect match with the target mRNA, leads to cleavage of the mRNA, again leading to the same result, that is no production of protein from the given or the target mRNA, and hence there is a gene silencing. Gene silencing refers to that there is a gene for a protein but no protein is being produced at the ribosomal level. There is another term called small interfering RNA. These are usually synthetic microRNAs. They are prepared in the laboratories and their main function is to cause knockout of the gene. They have similar activity like the microRNAs. When they are inserted into the cell, they interact with the dicer enzyme which cuts them into two pieces and then it acts similar to the microRNA, thus causing gene silencing. Hence the name gene knockout. The small interfering RNAs have also been successfully used for cancer therapy. You will come across frequently a term called mutation. A mutation is defined as a permanent change in the DNA. There are different types of mutations. Mutations can occur in the coding sequence. They can be point mutations. Point mutation means only one nucleotide is changed at a given point. Mutation can also occur within non-coding sequences. Mutations can lead to deletions and insertions. Deletions means small portion or a single nucleotide or many nucleotides in sequence are removed from the DNA, or insertion means single nucleotide or a number of nucleotides are inserted at a particular point, both of which will alter the reading frame of the DNA. So they are also known as frame shift mutations. Then we have a certain type of mutation known as the trinucleotide repeat mutations. In the DNA there are long chain sequences of trinucleotide repeats. For example, AGU, AGT, you can have a repeated sequence of AGT, AGT, AGT about 20 to 100 times repeat. These are known as trinucleotide repeat mutations. We shall talk more about these type of mutations and the diseases arising from them in the subsequent lectures. Few other things we need to know, but before that I must tell you that not all mutations are heritable. That means not all the change in DNA which occur in the body can be inherited. Only those changes which occur in the germ cells can be passed on to the next generation, because mutations can also be acquired. You go out in the sun, you get exposed to UV rays, you can have a mutation in the cell of the skin, but that mutation is not going to be transferred to your children, because these mutations are in the somatic cells and they are localized. They are not in your germ cells, right? So not all mutations are heritable. Secondly, some mutations are lethal. That mutation, when occurs, kills the cell. So the cells do not survive. So if the cells do not survive, then there is no way that they can transmit the disease. So only those mutations which are non-lethal and occur in the germ cells can be transmitted to the next generation. Then there are certain terms you should be acquainted with. Hereditary. Hereditary simply refers to disease which are heritable, which can be inherited from the parents and then which can be passed on to the next generation. Familial. Familial is synonymous with hereditary. Literally it means the disease which runs in family, and that mostly are hereditary diseases. Congenital means a disease which is present since birth. Now please note here that not all congenital diseases are genetic or hereditary, and not all hereditary diseases are congenital. For example, syphilis is a disease which can occur to the fetus in the uterus. So the fetus, when it is born with the disease. Such condition is known as congenital syphilis. But syphilis is an infective disorder caused by a bacteria. It is not a genetic disorder. So congenital diseases may not be genetic or hereditary. Similarly, there are many hereditary diseases which occur or which manifest later in life. They do not manifest at birth. So those diseases are hereditary but not congenital. So congenital and hereditary should not be used synonymously. They are two different words. You are also aware of these terminologies: autosomal disorders, X-linked disorders. Some disorders are dominant and some are recessive. Codominance, when both the alleles when present, they will express simultaneously. In human body, the blood group antigens are codominant. The human leukocyte antigen or HLA antigen, they are also examples of codominant genes. Pleiotropism, single gene, many effects. These are diseases where there is a mutation of a single gene, but they lead to varied manifestation in the person. For example, in sickle cell anemia. Sickle cell anemia is a hemoglobin disorder where the RBCs become sickled, but they lead to variety of other symptoms also. Apart from hemolysis, there are other symptoms. Genetic heterogeneity is the term which is reverse of pleiotropism. It means single effect, many genes. For example, hereditary deafness. There are many genes at different chromosomes, the mutation of which can lead to a single outcome, that is deafness. So such diseases are known as genetic heterogeneity. Dominant negative. This is a dominant disorder where the product of the gene impairs the function of the product of the normal gene. You know, there are always two alleles for a particular character. If one allele is abnormal and it is dominant and it produces a protein which interferes with the function of the other allele which is normal and a disease is produced, it is known as a dominant negative kind of inheritance. The example being osteogenesis imperfecta. Sometimes mutation lead to loss of function. For example, a mutation in the G6PD enzyme leads to inactivation of the enzyme and hemolysis. So these are called loss of function. Sometimes mutation lead to gain of function mutation. That mutation leads to excessive production of a protein or production of an abnormal or altered protein with a different functionality. The example of which is a Huntington's disease. We'll read about all these diseases later on. The difference between autosomal dominant and recessive disorder is sometimes asked. You have already this knowledge from your +2 science classes. This is just a repetition and revision of the same. A autosomal dominant disorder manifests in the heterozygous state, whereas the recessive disorder can only manifest in the homozygous state. Autosomal dominant disorder, you will find at least one of the parent is affected, but sometimes you will see that none of the parents is affected but the children is affected. This usually happens when it is a new mutation arising within the one of the parent germ cells of one of the parent. Then the parent will not manifest the disease, but he will pass on the mutated gene to the next generation, and the next generation will manifest the disease. Autosomal dominant disorders are characterized by variation or variable penetrance and expressivity. Now, penetrance is a term which means, for example, if 10 persons have that abnormal gene and only 5% express the disease, then the penetrance will be said to be 50%. So, autosomal dominant disorders usually have a variable penetrance. Expressivity means if 2 persons have the same mutation, they may express different symptoms and traits of the disease. One may have one type of symptoms, the other may have other type of symptoms, a third person may have combination of both the symptoms. So this is known as a variable expressivity of the disease, which is usually a feature of autosomal dominant disorder, whereas autosomal recessive disorder usually show a complete penetrance and a uniform expression of the disease. Many times autosomal dominant disorder do not occur in the childhood, but they appear later. So there is a delayed onset, whereas recessive disorders usually have a very early onset. Early in the childhood they manifest the disease. The autosomal dominant disorder usually are seen to affect the receptors and structural proteins of the body. Receptors, for example, the LDL receptor, which leads to familial hypercholesterolemia, is an example of an autosomal dominant disorder, whereas the structural proteins such as fibrillin, when affected, leads to a disease known as Marfan syndrome. We will talk in detail about Marfan syndrome in the subsequent lecture. Recessive disorders usually affect enzymes, for example, phenylketonuria or the lysosomal storage disorders. These are all enzyme disorders. Please remember that all hemoglobin disorders such as sickle cell anemia, hemoglobin D disorder, thalassemias, all are autosomal recessive disorders. Pedigree analysis is a method to know whether a particular disease is autosomal recessive or dominant. Comes the X-linked recessive inheritance. These genes are present on the X chromosome, and we all know that X-linked recessive disorder is characterized by a few things such as males are affected and females are carrier. As you can see in this diagram, two common scenarios of X-linked recessive disorders are depicted. On the left hand side, the mother is a carrier and the father is unaffected. In this scenario, the X chromosome from the mother is transmitted to the boy and the boy is affected. The X chromosome is also transmitted to the girl, but because the X chromosome of father is also transmitted to the girl, so a normal X and an abnormal X are present in the girl and the girl becomes a carrier. So in any given pregnancy, there is a 50% chance of a boy being affected by the disease, and there is a 50% chance that if a girl is born, she will become a carrier. On the right hand side, the scenario is that the father is affected. That means his X chromosome is abnormal, he has a normal Y. The mother is unaffected. That means both her X chromosomes are normal. The boy of the next generation will get the X chromosome from the mother, so all boys will be normal. Whereas the girl may get a X chromosome from the father as well as from the mother. So all girls which are born in the next generation will be carriers. They will be carrier because they have one normal X chromosome from the mother. Please spend some time on these pedigree charts and you will understand. X-linked dominant disorders are very rare. The best example is vitamin D resistant rickets. In this picture also, two possible scenarios are depicted. In the left hand side, the father is affected and mother is normal. So the son will be unaffected, as the son will get the X chromosome, normal X chromosome from her mother. The son will be normal, whereas the female gets one X chromosome from the father and one X chromosome from the mother. The X chromosome from the father is abnormal, and since this is a dominant disorder, the daughter will always be affected. In the other scenario, the father is unaffected and the mother is affected. That means the mother harbors either a single or both X chromosomes are abnormal. So there is a 50-50% chance that the girl will be affected or the boy will be affected. That is, in each pregnancy, whether it's a girl or boy, there is a 50% chance that the offspring will be affected by the disease. This is all for now, but in subsequent classes we'll discuss about various Mendelian disorders and the different type of genetic disorders. Thank you.